How's that? Can you hear me now? Okay, well, I just have a couple of general announcements here before we get started. A few people have written about whether or not the readings that are posted online for this part of the course apply to the 8th edition, the previous edition of Campbell. And for this part of the course, they do exactly, so there's no reason to post a different set of readings. There's some very minor changes, but nothing too significant. So if you have the 8th edition, just go ahead and read the pages indicated online. And also, really importantly, on Monday, be sure that you manage to set your clock back, or else you'll be here an hour before I will. And it's a pretty bad place to get your last hour of sleep. So be sure to do that. That's Sunday. And so today we're going to continue with fungi. Wrap that up quickly. Last time near the end, I spoke a bit about the ecological importance of fungi and talked about their importance as decomposers, especially of wood and plant material, and also about their role in mutualisms with plants, both with the roots as mycorrhizae and with the leaves as endophytic fungi that are really important to the success of land plants. And as we'll talk about maybe a little bit later, it's arguable whether land plants could have become established without fungi. And also we talked about lichens, which are really important in primary ecological succession, which are an association between a fungus and a unicellular photosynthetic organism, a green alga or a blue-green bacteria. So the last major topic of ecological importance with regard to fungi is their importance as disease-causing organisms. And there are about 30,000 fungal species that could be characterized as either parasites or pathogens. So a huge number, about 30% of fungal species. I guess fortunately for us, most of these affect plants, although they affect plants that are ecologically and economically really important. I mentioned some tree diseases earlier. Some of the ones that have been most devastating, and this tends to be true for diseases in general, are the result of introduction of the disease-causing organism outside of its native range. And for example, between continents, especially problematical, the Dutch elm disease, which has wiped out a lot of American elms in the east, and the chestnut blight, which has pretty much driven the American chestnut to biological extinction. They continue to stump sprout after over 100 years of decimation, but they don't reach maturity. And so these are diseases that came in from other parts of the world and involve fungi, ascomycetes in particular in this case. But there are other diseases of crop plants, both trees and grains, that are really important. And we'll talk about one of those in a moment. So fungi also affect animals, of course, and not nearly as many fungal species or animal parasites, but there's some really notable ones. And one that I'm going to show you in particular, I should have shown you on Halloween because it's particularly gruesome, but it involves insects. And insects, as you know, are sufficiently prolific that their reproduction can stay ahead of mortality, even if mortality is pretty high. I mean, a parasite or disease-causing organism is at ultimately a disadvantage if it kills its host, usually, but in the case of insects, they can get away with it. And there's a whole clade of fungi in the ascomycetes called cordyceps that have diversified with insects, about 400 species of these very specific pathogens. And I'm going to show you an example here. And they actually not only infect the insect, but they change its behavior to their benefit. And they basically replace the insect's tissues with their own mycelial tissues. And this is a case at the last end of the stage of infection for an ant here. It's now crawling up some vegetation. That's a stereotypic behavior for this particular species of ant and its associated cordyceps fungal pathogen. And now you can see it's crawled up and it's clung to the piece of vegetation, which is stereotypic behavior. And now it's dying or it's dead. And the fungus is starting to produce a fruiting body out of the back of its head. It's already completely consumed its tissues. And now you can see the fruiting body there forming. This is time lapse. This isn't in real time. But that's the sort of thing that this whole group has done. And you can see there's some interesting diversity here of different species of ants just in this one clade of cordyceps. And you can see different mushrooms that have different morphologies. So this is a group that has capitalized on ant parasitism. So that's a Halloween image, I think, a little late for the holiday, but that's an unusual case. We'll get back to diseases of humans in a moment because we're going to talk about the direct importance of fungi to humans, and that also has to do with disease agents. But really importantly for us, in addition to their ecological services, are the importance of fungi as food. And some of the things we take for granted in life and that make life pleasurable, like alcoholic beverages, wine, beer, liquor, results of alcohol fermentation involving Saccharomyces yeast. And so yeasts are involved in that, of course. And also in the leavening of bread, the rising of bread, we have to use yeast as well, also in the genus Saccharomyces. And in some cheeses, Roquefort cheese, blue cheese, that's infected with a type of ascomycete that's multicellular, not a unicellular yeast, that imparts those flavors and coloration to those particular cheeses. So there's a lot to be thankful for about the fungi as far as our diets and lifestyles go. And there's also, of course, a lot of fungi that are edible. We already talked about morels and truffles. I just mentioned them as ascomycetes. But a lot of basidiomycetes have edible fruiting bbodies, mushrooms that we eat all the time. None of these quite rise to the same level of economic value as some of the truffles, but they're really important in diets, both of Western diets and also a lot of all over the world, and many indigenous peoples collect mushrooms as part of their main diet. Some of these are important for fighting cancer, like shiitake, also really a delicious mushroom, but great, its extract's very effective or somewhat effective in cancer treatment. There's a lot of research going on with that. But there is a flip side to fungal ingestion, and that is that some fungi that are deadly poisonous look somewhat similar to those that are choice edibles. And people that gather mushrooms, of course, have had a very strong reason to tell the differences between these kinds of edible and poisonous fungi. But people that move from different parts of the world, recent immigrants especially, can be fooled by the native mushroom flora and can be poisoned seriously. And it's a real problem. And for example, North America, we have mushrooms that are deadly poisonous. This is true in other parts of the world too. Members of the genus Amanita can be really deadly and cause liver and kidney failure and certain death unless you're treated immediately or get a transplant of those organs. This is a case shown here. We can see these two mushrooms look really similar. The one on the right, a paddy straw mushroom, is a choice edible Southeast Asia. And some recent immigrants that have been collecting these confuse Amanita phalloides, one of the other Amanitas, which are one of the world's most dangerous mushrooms, with these and result in mushroom poisoning. So if you're a mycologist by trade, and university mycologists are often on 24-hour call, at poison centers, public service they provide to identify the mushroom that the person ate that caused them to have a complete health crisis. So this is a potential hazard. You can tell these kinds of mushrooms apart by spore prints. Take the cap off and get a spore print, and the spore print colors are different in many cases between edible and poisonous mushrooms, but that's not always reliable, and you have to be super careful. A really insidious type of fungal poisoning is known as ergotism, and this is something that fortunately doesn't happen much anymore, but it killed thousands of people back in the Middle Ages, and it happens when people mill grain, rye that is, type of grain, of course, which is infected with a fungus, an ascomycete, Claviceps purpurea, the scientific name is not important. In any case, that's a fungus you can see on the right there. Those slug-like dark structures are the fungus. And when it's milled with the grain, the poisons it produces are stable, and people that eat products from that grain can come down with symptoms ranging from gangrene to seizures, hallucinations, and temporary insanity. One of the compounds that's been isolated from these fungi is lysergic acid, which is, of course, the natural substance from which LSD is made. And there are much nastier substances in this fungus as well that cause some of these other fatal symptoms. But it's also had potentially indirect causes of mortality. If we go back to the Salem witch trials in colonial America, there were individuals you're probably aware of that were hung, executed as witches, and there's some suspicion, not proven, but possible that some of these individuals may have been victims of ergotism, were showing symptoms of this poisoning, and indirectly led to their deaths by execution. So this is still some controversial interpretation of that, but it's an interesting one. All right, so disease-causing fungi are probably familiar to all of you. Anyone that suffered athlete's foot or ringworm or jock itch, these are all types of ascomycete poisoning, or ascomycete, we call them diseases. They're easily treatable and temporary afflictions. But these fungal diseases that affect people are no joke. If you're an immunosuppressed person, say having like full-blown AIDS cases can be killed by fungal infections and often are. I could show you some photographs of human victims of some bad fungal infections that are fatal, but when these were shown in a previous class, one of the students passed out, and I don't want to have that happen this morning. So if you want to see any of that, you can come to office hours and I could show you. But with the disclaimer that I'm not responsible for your reaction to that. But basically, if you saw what happened with the ant in the previous little movie clip, it's not that far off from what can happen with people. Mycelium can pretty much take over, and fungal fruiting bodies can start to emerge out of the face and various places. Pretty hideous. So that would have been the real Halloween image, being way beyond hideous. Okay, but on balance, fungi are really important as disease-preventing organisms, and millions of people owe their lives to antibiotics. The understanding in the 1940s that penicillin could be used to treat bacterial infections was a gargantuan breakthrough in medicine, and antibiotics, of course, are still absolutely critical. The way that penicillin works, well, you can see, first of all, a little colony of penicillin up the top of that petri dish, and notice that zone of death around the lower side of that circular colony where the plated out bacterial culture is dead. It's clear in that area. And the way that this works is that the penicillin interferes with the peptidoglycan in the cell walls of bacteria. So the fungal cell walls, remember, are made of chitin, and we don't have cell walls, so we're free and clear with the use of this kind of treatment. But the bacteria are constantly needing cross-linkages between the peptidoglycan and their cell walls, and this interferes with those cross-linkages, so it's a really effective antibiotic. And penicillin, you're probably more familiar with in nature as decomposers, for example, fruit, like you can see there. All right, so any questions about fungi before we move on? We're going to switch gears now. That's pretty much all for fungi. And we're going to move on to photosynthetic organisms. And before getting to land plants, we're going to talk about this great diversity of organisms we call algae, which are photosynthetic, and they're also polyphyletic, as we already talked about briefly. The different groups of organisms called algae are circled in orange there. And you can see they're scattered over the tree of life. Land plants are closely related to some, but not all of them. And so it's really quite a strange thing to think about photosynthesis, oxygen-generating plant-like photosynthesis, occurring in so many disparate lineages of eukaryotes and even one prokaryotic lineage. And we'll get to that in a moment, but maybe not surprisingly, given that phylogenetic diversity, algae, which I have in quotes because it's not a monophyletic group, it's not a clade, span a huge range of forms, lifestyles, ecology, and chemistry as well. They range from unicellular organisms like the diatom in the lower left here to huge organisms that basically form forests off the coast, very notably here in California. And we'll show some more about that in a moment. So a lot of diversity here, all photosynthetic. And here you can see a simplified diagram of the 5 major clades of eukaryotes. This doesn't include the bacteria, which also include an algal group, the cyanobacteria. But in dark background and white lettering here, you can see different photosynthetic organisms again. So basically 4 of the 5 major clades of eukaryotes have these photosynthetic organisms that we call algae, except for this one, the epistoconts. The name's not important, but that's the clade in blue there that includes the fungi, which are also botanical organisms, but not photosynthetic, as well as the slime molds and other amoebozoans and animals. So there aren't any photosynthetic organisms in that clade, but in the others. So the question naturally arises, how did photosynthesis, very, very similar photosynthesis, evolve in so many different groups? And to understand that, you have to really understand the endosymbiotic theory, which Lynn Margulis proposed, and for which she was widely ridiculed for many years. She should have received the Nobel Prize, but never did. She died last year. And the basic theory has been pretty well demonstrated now as being accurate with regard to the evolution of mitochondria and plastids, which are the most significant organelles, the ones we're going to talk about here. So to really understand this idea, endosymbiotic really means living inside another organism. Remember, symbiosis is living in close proximity. Endosymbiosis means living inside. And so basically this theory in regard to the components of it that we're interested in for the purposes of this course have to do with the inclusion within an ancestral eukaryote of bacteria that came to be inside of eukaryotes and almost certainly engulfed by an ancestral eukaryote. You might ask, well, how could it engulf it? One of the early innovations of eukaryotes, even before they acquired mitochondria or plastids, must have been the cytoskeleton. So the cellular skeleton, which includes microfibrils and microtubules that move things around inside the cell and allow the cell itself to move. Think of the amoebozoans like the slime molds. And also that allow eukaryotes, at least some of them, to actually ingest prey items that are larger than those that can just diffuse through their membranes. And so by enabling the cytoskeleton, allowed them to be able to ingest larger prey items. And almost certainly the first of the two major types of organelles to be ingested was the mitochondrian, which is what's being shown, was the organism that became the mitochondrian. That's what's being shown here. Proteobacterium that resided for, it may have been almost certainly a situation where the cell was ingesting this as a source of food, but it came to live inside the eukaryote, which might sound a little far-fetched, but they're great examples of these kinds of endosymbioses happening within modern time, and people have observed this sort of thing happening where an organism gets ingested and it's not digested, and it lives there for some time, sometimes as a parasite. But in this case, it proved to be beneficial, and eventually, over evolutionary time, there were gene exchanges between the mitochondria and the nucleus that made these organisms inextricable, where they couldn't live without each other. And so here's a little cartoon starting at the top where you see an early eukaryote before we even have a nucleus. And the nucleus is not something that's thought to have been acquired by engulfing of another organism. The nuclear chromosomes are basic to the survival of eukaryotes. And the membranes that surround the nucleus are similar to those of the endoplasmic reticulum and the inner membranes of the cell, and were probably elaborations from the perimeter of the cell towards the center that ultimately resulted in a nuclear envelope. But eventually we have the origin of the mitochondrion here, where you can see a proteobacterium being acquired and eventually being completely incorporated into the cell as part of the cell. But in some lineages we also, So the idea is that it's a great question. The early eukaryotes were probably anaerobic, and so they were probably engaging in anaerobic respiration, which doesn't require mitochondria. But and it also, to an anaerobic organism, oxygen is often toxic. I mean, they can't even survive in an oxygen-rich environment. So by acquiring the mitochondrion, it may allow them to, survive in this increasingly oxygen-rich environment as a result of cyanobacterial activity. So it was, the fact that every eukaryote in the world now has mitochondria, nobody's discovered one that doesn't, is a pretty good sign that acquiring mitochondria was a key innovation. And without acquiring mitochondria, the kind of world that the cyanobacteria, by producing all this oxygen through photosynthesis, created would have been the end of the eukaryotes if they hadn't acquired the ability to handle oxygen. And so acquiring an aerobic organism, a proteobacterium, was probably crucial to their survival, given that none of them exist now without it. So that's a great question. And so in some cases, not all cases, we're on this line here, one of these lines that didn't acquire chloroplasts, but some lineages did, and that's the evidence suggests that was a cyanobacterium that was acquired in the same way and incorporated. Get to that evidence in a moment. So the endosymbiotic theory then leads to the conclusion that cooperation between eukaryotes and prokaryotes, really distantly related organisms, was key to evolution on Earth. And this compelling idea at the time was proposed, but it was a tough sell. It's a radical idea, as you can imagine, and radical ideas tend to get a cool reception at first. But here's some of the evidence that pretty well clinches it. The first thing, and one of the obvious things that was first noticed, is that mitochondria and plastids, such as chloroplasts, are about the same size as bacteria, or the bacteria they're thought to be closely related to, and they have similar internal structure as well. You can see pretty similar overall ultrastructure there. Also, something really interesting is that both mitochondria and plastids reproduce by binary fission, as you can see going on here, which is the way in which prokaryotes reproduce. And if you destroy the plastids inside of a cell that had plastids, that's the end of the plastids in that line. There are some organisms that are photosynthetic that can also ingest other organisms. I'll show you one of those in a moment. But it's not, the plastids are not encoded by the nucleus, nuclear DNA. They're something that only persists in cells because they can reproduce on their own, and the same with mitochondria. Not only that, but if you look inside of these organelles within eukaryotes, mitochondria, and the plastids, they have ribosomes that are different than the ribosomes that are out in the cytoplasm of that eukaryotic cell. They're different in size and in structure. So a ribosome, remember, is a unit that's complex of ribosomal RNA plus protein subunits that are crucial to the translation of messenger RNAs into polypeptides or proteins. So they're crucial in translation. And the eukaryotic cytoplasm has a different kind of ribosome than the ribosomes that we find inside of plastids and inside of mitochondria. And those ribosomes inside plastids and mitochondria are very much like the ones that we see in bacteria. Okay, so that's pretty interesting too. And even more interesting is that if you use bacteriostatic antibiotics against eukaryotes, these are antibiotics that interfere with bacterial growth by affecting their ribosomes. This also affects the ribosomes of the organelles of eukaryotes, but not the ribosomes of the cytoplasm of eukaryotes. So these ribosomes inside of mitochondria and plastids are not only structurally similar to those of